There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
For some 3 decades, scientists around the world have been struggling with what causes Alzheimer's disease. How does it begin? How does it unfold? In the brain, synapses transmit information from one neuron to the other. And this burst of blue light represents the synapse communicating. But in Alzheimer's disease, it goes cold. You see the synapse stop firing, and the information is no longer transmitted, and the nerve cells die out. In the clinic, we see synaptic failure. So these clocks are examples of what I ask my patients to do. Can you draw a clock, the face of a clock, and put the hands in so it reads 4:45? And my patients do different things. Here, this patient puts in the hands incorrectly for 4:45. This more advanced Alzheimer patient can only put a simple box around the numbers 4 and 5. This patient took my command literally and thought that I asked her to draw the face of a clock. And so it looks like a face. When I repeated the command and said, no, I mean really what a clock looks like, and put in the hands, she did it correctly. This patient has such severe Alzheimer's disease that she can't really put a clock together. So this is a high-power view of a little part of the brain of a patient whom I followed during life. This was a 69-year-old gentleman who had a family history of Alzheimer's and died after about 10 years of disease. And when I took the brain out and made this picture, we saw 2 spherical deposits of protein called amyloid plaques. These are big conglomerations of protein that shouldn't be there. Plaque, it's made up of literally hundreds of thousands or millions of a little protein that we call amyloid beta protein, sometimes called beta amyloid or A beta. And in this diagram, there's a large protein that we call amyloid parent protein, or APP. And the A beta region of it is colored yellow. And the question is, how does just this little area actually form these plaques in my patient's brain? The orange abnormalities are just the yellow part of this diagram built up into a huge clump. We've now figured out that 2 different enzymes cut the parent protein to release the amyloid beta region in yellow, and it floats away outside the nerve cell. Well, what actually happens is that if the amyloid beta from this molecule meets the amyloid beta from another amyloid parent protein, they can stick together. And that sticking process forms a dimer or doublet. We use the term dimer for 2, trimer for 3, and collectively we call these things oligomers. This is a process that we've come to believe is at the heart of the Alzheimer memory failure. The oligomers can float into a synapse, which is the connection between this big nerve cell on the right and this big nerve cell on the left. If we magnify this synapse, You can see A-beta oligomers float into the synapse, and they can short-circuit nerve cells important for memory. The information is no longer transmitted, and the nerve cells die out. Now, after a while, these dimers and trimers build up and stick to each other to get bigger and bigger structures. And that, ultimately, we believe, forms the amyloid plaque, which Alzheimer first called attention to. As far as we can tell from experiments, this large plaque may be less toxic because it can't float into the synapses. Alzheimer's disease is an imbalance between the production and the removal of this amyloid beta protein. And if you don't remove it properly, or if you overproduce it, it builds up in your brain. It can form oligomers, which can short-circuit nerve cells. We want to block that process. We have several ideas, and the good news is that because Alzheimer's is a terribly common problem, many scientists, both in academia and in industry, are trying to find things that block it. And the great news is that we're very close now to having evidence that certain treatments that lower A-beta might actually slow down the dementia.
Dennis and I designed an experiment. We're using this treatment to see whether we can stimulate the body's own white blood cells to clear out the amyloid from the brain. The white blood cells are programmed to fight off infection and to keep the body under control, to keep all the inflammation under control. If you cut yourself and you have, I don't know, a scar or something, cells come and clean it up. And so could we use the immune system to clean up the amyloid in the brain?
The local immune system doesn't do it well enough. Otherwise, Alzheimer's wouldn't be a progressive disease. But it tries to do it. And I think some of Howard's most recent ideas about how to stimulate the local immune system are really very exciting and seem to work beautifully in mouse models.
We're trying to develop a nasal vaccine for Alzheimer's. These are mice that for 8 months have been getting a nasal treatment. Here's some of the results that I can go over with Dennis. We just published these.
So what we're looking at here is a picture of the slice of a mouse brain where the green is the actual amyloid. And in this animal that's been treated with the nasal vaccine, there is a dramatic reduction in that. And this is, of course, what we're hoping for even in humans, ultimately. So if mice were men and women, this would represent, obviously, a dramatic removal of amyloid. The administration of the vaccine through the nasal cavity is something that Howard had himself worked on intensively and pioneered in the area of multiple sclerosis. This is a little bit of an unusual way, as we think of vaccines, when we think of an injection type.
By giving it nasally, we're actually stimulating the immune system in the brain better. We are always eating food, we're breathing in things, so that the body is used to taking things in and not reacting in a negative way. So we're trying to use the body's natural immune system, the natural way we're exposed to things to treat a disease. And we're predicting that if we can do it, it won't be as toxic, it'll be easy to give. In fact, this nasal vaccine that we're working on is used, some of the components are used now in people for influenza. So we're taking advantage of something that's already being tested in people.
I think the understanding just in the last decade, but especially even the last 2 or 3 years, is phenomenal. And so I think we are very much on a roll. We've learned enough that while there's plenty of T's that are not crossed, plenty of I's that are not dotted, we're just about there. We have treatments that are in hand, and we already have clinical trial results that suggest that we're on the right track. Now, there's still skepticism until everyone who can get Alzheimer's disease doesn't get it anymore or gets it much less severely. We will not be satisfied.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
The history of modern medicine is really tracked back to around 500 years ago when people decided that it might be a good idea to crack open the body and actually see where the disease is. If someone is bilious, is his liver really affected? If someone is demented, is his brain affected? And it's not that long ago, but that really gave rise to this idea that we actually have to see the disease to understand it. Alzheimer's was a German neuropathologist. His use of novel staining techniques in dead brain under the microscope allowed him for the first time to see this constellation of amyloid plaques, neurofibrillary tangles, and it ultimately justified calling this disease Alzheimer's disease. Now, it's interesting to point out that if you look at textbooks of neurology, psychiatry, medicine, for decades thereafter, into the 30s, 40s, 50s, there's nary a mention of Alzheimer's disease.
Serious and focused research on Alzheimer's disease really began when the National Institute of Aging and a variety of other researchers, brain researchers, realized that what we had been calling dementia or senile dementia or hardening of the arteries or arteriosclerosis in late life that everyone just called dementia, actually in the vast majority of cases was Alzheimer's disease. Which before this we had thought had been this very uncommon disorder that occurred in your 40s or 50s in rare cases.
Once the field and the world recognized that this relatively common entity of senility, quote unquote, was a true disease, then it really justified a concerted research effort. In the 80s, we were able to isolate the main building block, the main peptide that makes up one of the core features that Alzheimer's himself saw, namely amyloid plaques. And with that information, we were able to identify the parent protein that produces amyloid, which is called amyloid precursor protein, or APP.
When we started, no one knew anything about what plaques or tangles, the things we see in the brain, were made of. We now know that exactly the proteins that they're made of. We understand the pathology and how the pathology developed at the microscopic level. We understand the processes that take us to amyloid plaques and how they damage nerve cells. And we also understand the very earliest aspects of what happens inside the cell that leads to neurofibrillary tangles in the brain in Alzheimer's.
In the 1980s, we made tremendous progress in first, understanding what the disease represents, second, how to diagnose it, and three, to understand something about how it begins in the brain, how it changes brain cells, damages brain cells, and results in its clinical phase, dementia, loss of memory and thinking ability. Tremendous number of tools have helped this remarkable advance. In 1991, the first gene mutation that causes Alzheimer's disease in an inherited fashion in a particular family was reported, and it happened to be in that amyloid beta gene. So within 10 years, the discovery of an abnormal protein, amyloid beta, in excess amount in Alzheimer's brain led to the discovery of the gene producing it, and then the gene, when mutated, causing the disease.
It's worth explaining why that's so important, even though early onset Alzheimer's disease is so rare. The reason is because until that point, there was really no idea, no clue as to whether amyloid plaques or neurofibrillary tangles, the hallmark features of Alzheimer's, were really causing Alzheimer's disease. And so once amyloid mutations were found, it argued very forcefully that, in fact, amyloid is really one of the main causes of Alzheimer's disease.
There are only 200 to 300 families in the world who have these kinds of mutations that lead to familial early onset disease. They are valuable far out of proportion to their numbers because they're where all of the models, our mouse models that we can use to look to find drugs, all come from the genes in its families.
You can take that mutated gene and put it in a mouse. Take the gene that causes Alzheimer's disease, put it in the mouse. The mouse develops a form of Alzheimer's. It's not classic Alzheimer's disease, but it certainly develops amyloid plaques. We began in the 1980s with trying to just understand what the disease represents. In the 1990s, we began to get some treatments that helped the symptoms of the disease, but now in this decade, we understand that the illness, in order to really be effectively treated, is going to require agents that get at the underlying mechanisms of the disease. Can we make antibodies that target amyloid in the brain? Lo and behold, in at least those transgenic mice, when a vaccination approach was employed targeting that amyloid by antibodies, it seemed to really have a remarkable effect. It removed those amyloid deposits. I never would have thought that. So then that led to a whole host of immune-based, antibody-based therapies. Not all vaccination, some infusing the antibodies. And there is great hope that they will be effective in clearing out the amyloid in the brains of people with Alzheimer's disease.
If you think about all the successes that we've made in medicine, they've really come on the heels of being able to visualize the problem, whether it's cancer, heart disease, various problems of the digestive tract, and in fact, brain disorders, stroke, MS, multiple sclerosis, tumors. One of the great challenges of Alzheimer's disease is the fact that until recently it's been relatively invisible to our technologies, and I think that's why it's been such a great challenge. But that, I think, is shifting now.
Pittsburgh Compound B, or PIB as it's known, is a radioactive tracer that you give in a vein that, when you do an image, shows you the amyloid plaques that are in the brain of people with Alzheimer's disease. It will absolutely show you plaques in a living human.
I think it's going to revolutionize both how we do research, as well, if we continue to validate this technology of imaging brain lesions, it will eventually be very helpful for general practice in diagnosing the disease.
Okay, you're good to go.
We now have optimized a MRI-based technology that allows us to really capture the earliest stages of the disease. We now know very clearly that there's an earlier stage of Alzheimer's that can be called the cell sickness stage, where the cells are there, but they're just malfunctioning. The good news, though, is that if we can capture that stage of the disease, it really, I think, offers a lot of hope for better treatment. I think it's obvious to say that it's easier to treat a sick cell than a dead cell. And in fact, now as we've finally entered in an era of drug trials for Alzheimer's disease, I think what the future is going to bode is an attempt to really try to capture people who have this cell sickness stage, as difficult as it is to detect, because though it's going to be that stage of the disease that's going to potentially benefit most from treatment.
We've made a paradigm shift. At least our research is dedicated to the proposition that we don't just want to wait till the disease is fully manifested in the dementia stage, but we want to make a commitment to seeing if we can't prevent the illness.
We will find a drug that slows things significantly, and then we'll figure out what it does and what it doesn't do and make a better drug. And then we'll add two drugs together. because many of these drugs that attack different aspects of the disease could be used together. And we'll do the same thing that the oncologists, the cancer docs, did with lymphoma and leukemia. We'll slow down this disease process, an inch at a time, with luck, with a good drug, a foot at a time. But we'll get there. And I can say that in the 80s, a lot of the people who were senior people in the field thought, there's no way you can ever help this disorder. There's no way you can find a way to attack these things. We don't know what they are. So in 20 years, we've come to the point where our expectation is that we will find things that will slow things down. Our only sorrow about this is we can't do it instantly. The nature of the disease is such that it takes time to prove that the medications work.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
A very common question that we get is, how much forgetfulness is too much? I think the forgetfulness that many of us experience as we age, where are my car keys? I come home, I flip them on the counter, and I can't find them 5 minutes later. Or where are my reading glasses? Or where's that checkbook? I don't think that kind of incidental forgetfulness is all too serious. I think that's very, very common. But when we get into the arena of I'm now forgetting the types of events that I used to remember fairly easily. So when you really try to remember something and you still forget it, that becomes a little more problematical. May not be Alzheimer's disease, but I think it warrants attention. You should mention that to your personal physician and say, should I be concerned about this? Should I be pursuing this further? There's a period of time during which people will be sort of partially symptomatic. People are slightly more forgetful than they used to be, and perhaps more forgetful than they ought to be. And that period of time we've characterized with the term mild cognitive impairment.
This is myself and ARPA. Well, this picture is of my father when he was probably 50 years old, and a big husky man, healthy, and these pictures I think kind of show how dementia progressed with him, and that scares me. I don't actually know for certain that it was Alzheimer's. To the best of my knowledge, at first they thought it was a different dementia. He knew that he was having a problem, and he said, the way he put it, Bob, I didn't ever want you to see me this way. Someone that when the other thing that bothers me right now, my train of thought just left. And when that happens, then I'm more aware than ever that I'm not invincible. We go another block and come in that way? You get tired?
Our work on mild cognitive impairment is designed to try to pick up people with the very, very earliest stages of forgetfulness. It may or may not mean that the person is going on to develop Alzheimer's disease, but may have another type of cognitive impairment that is evolving over time. Other dementias, such as frontotemporal dementia, dementia with Lewy bodies, vascular dementia, are also outcomes that do occur in aging. So we need to distinguish among those various types of dementing illnesses.
Dr. Peterson.
Good morning, Mr. Emmer.
Good morning.
How you been doing?
I think in some areas quite well, and others maybe not so good. But
okay. What in particular has been not so good?
I think maybe while I'll be talking to you, and specifically and everything going fine, and I start to tell you something, and all of a sudden I lost my train of thought, and that bothers me because.
And that's different from
That's different. It never,
a year ago, 2 years ago, 3 years ago, something of that
Yes,
nature.
I think it is.
One issue that we've been following over the years is your sleeping habits. Tell us about what kinds of sleeping problems you've been having.
Every night I put that chair beside my bed because sometimes I get some violent dreams or something, and I might land on the floor. Or what it really scares me is if I would hit ARPA or something, and she says I never do.
I'm going to read a list of words. Listen carefully. When I'm done, you tell me as many of the words as you can in any order. Here we go. Doll, mirror, nail, sailor, heart, desert, face, letter, bed, machine, milk, helmet, music, horse, road.
Road, helmet, machine, horse, doll, mirror, nail.
There are different forms of mild cognitive impairment. Either a memory factor is involved so that the person has an amnestic form of mild cognitive impairment likely to go on to Alzheimer's disease, or they have a non-amnestic, meaning that they have difficulties in cognition, but it's not primarily memory. It might be attention, concentration, language, visuospatial skills.
I'll give you 4 blocks, and I'd like you to make it look like this. Tell me when you're finished.
Visuospatial skills refer to our ability to navigate in the environment or appreciate the visuospatial features of pictures and photographs and the like.
It's not as easy as it looks, is it? That's okay.
Mr. Emmert has what we call a non-amnestic form of mild cognitive impairment. We have found that these people tend to go on to other types of dementias. Again, not Alzheimer's disease, but maybe dementia with Lewy bodies. Sometimes people go on to develop frontotemporal dementia or vascular dementia. They have a different cognitive profile. In general, putting it all together, there is a slight decline from last year, but it's not too dramatic, and it's in multiple areas of function. Memory is impaired, but it's not the major area that's impaired. But the interesting feature, though, is that it's not an Alzheimer-type pattern. That is, your primary difficulties in thinking are with processing speed, paying attention to what's going on in the environment, and then this visual spatial. That, coupled with your history of the sleep disorders, makes us think that this might be on the road to a Lewy body-type disorder.
Lewy bodies, and that's a new term to me. I have not been exposed to that particular thing.
Lewy bodies are a form of a protein that gets deposited inside nerve cells, and again, they're characteristic of conditions such as Parkinson's disease. The interesting thing about Lewy bodies is they can be deposited in cells or nerve cells or brain cells in various regions in the brain. And sometimes they get deposited in the brain stem, the base of the brain. And that's a part of the brain that's involved in controlling sleeping, and in fact may relate directly to the specific symptoms that you are experiencing. That's why we hinge so much on your sleep disorder and the problem that you've been having for many years, because we think that this may be the very earliest feature of the Lewy body deposition in that part of the brain. And those can be treatable with medicines that we use for Parkinson's disease. We characterize dementia as sort of an umbrella term. While all of the dementias have similar characteristics with regard to impairments in thinking and affecting daily function, there are subtypes of dementia that are really quite different, and we can tease those apart clinically, and it corresponds to what's going on in the brain pathologically. Different types of proteins are being deposited, different proteins are deposited in different parts of the brain, and that displays itself differentially with respect to the clinical presentation. So, in Alzheimer's disease, the plaques and tangles form in the memory parts of the brain, and a person becomes forgetful. I think if a person gets the label of dementia, that's important, but I wouldn't stop there. I'd go the next step and try to find out what's causing the symptoms, what's causing the dementia, because it may very well dictate treatment and management down the road.
What are we going to trim?
See this?
I had trouble remembering last names, and
and I know the people by their first name, but I can remember the places and so forth. And
I think the short-term memory is fading faster.
yeah, I'm sure. And I was good in calculus and algebra and the different trigonometry and things of that nature with all the sine waves and curves and stuff. And then I got to the place where I was having trouble doing that.
Approximately 3 to 4 years ago, we made the diagnosis of mild cognitive impairment of the amnestic type in Mr. Braun, and we have followed him now every 6 to 12 months over the succeeding years. And what we've witnessed is a gradual decline in his memory function. Hi, Mr. Braun.
Hey, good morning.
Hi, good to see you.
Beautiful day out there.
Absolutely. Have a seat here. Thank you. Mr. Braun, you came to see us several years ago because you were having some memory difficulties.
I did call you by your first name, but gee, I couldn't remember who the last name was. And that's not good when you're meeting people in public and stuff like that.
You were telling me a couple of years ago you were helping your son build a house.
Yeah, usually I was the one that would be responsible for telling them what to do, but I got dependent upon him for all the measurements and putting everything together. Prior to that, I'd be in charge, but he basically took over with the...
So that was sort of a change in roles. Okay, now what we'd like to do is repeat some of our cognitive testing this morning, as you've done in the past, and then I'll visit with Mrs. Braun as well, and we'll kind of put it all together and see where we are at this point.
Sounds good.
Good?
Yep.
You've been aware of changes in him over the past months, year, or a couple of years. Can you share some of those thoughts with us?
He before he would think ahead. Now he thinks backwards. We'll plan this and this and this. Now it's what I did in the Boy Scout troop and what Fred Clausen did when he took us out to Hidden Falls, you know.
So a lot of reminiscing.
Right.
What's the highest level of education that you've completed?
I've got a master's degree in business.
Nice.
I graduated from the Naval Academy, and so I had a career in the Navy and then do the aerospace, Honeywell guidance packages and stuff for 10 years.
It's not uncommon in aging, and in particularly in Alzheimer's disease, that a person has very good recollection for old memories, but they may have difficulty remembering something that occurred recently. That may actually imply that they haven't formed that memory accurately in the first place. So it's a combination of impaired learning or encoding of new memories and retrieval of that information once it has been formed.
Well, now I'm going to read you a list of words. Listen carefully, and when I'm done, you just tell me back all the words you can remember. Doll, mirror, nail, sailor, heart, desert, face, letter, bed, machine, milk, helmet, music, horse, road.
Boy, I didn't get a story with that one. Let's see what I'm going to be. I don't get another shot at it.
Tell me all the words you do remember, even if it's just one.
Well, road was the last one,
Okay.
and I was trying to put a story together with it, and then didn't pan out. But
anything else?
Well, there was a road, and
When people start to forget information that they formerly would have remembered quite easily, it implies to us that there may be something going on in the brain that might be pathological. There might be a buildup of proteins in the brain that are now interfering with memory function, with paying attention, with finding the right words. So here's your MRI. The black areas in here are water spaces or cerebrospinal fluid spaces in the brain. That's normal, natural fluid that surrounds the brain. But to the extent the brain shrinks with age, those spaces tend to increase. So there's more black as the brain shrinks. There's probably more shrinkage there than we would like to see for aging. The memory testing, remembering the list of words and all that, again, your memory impairment was present. I think we're now getting into that gray zone where other things are becoming impaired a little bit. and your functional activities are becoming impaired a bit. So we may be at that threshold that this is now what we call clinical Alzheimer's disease. But this is a gray area. There's no sharp distinction. I don't have mild cognitive impairment one day and Alzheimer's disease the next day, but rather it's a gradual progression.
Right.
You may very well be eligible for participating in a clinical trial to evaluate the effectiveness of medications down the road. We can be fairly accurate with the clinical diagnosis of Alzheimer's disease. We can make some practical suggestions, lifestyle modifications. We can treat people with symptomatic medications that might help their memory a little bit. But the medications do not cure the disease. They don't stop the underlying disease process. Alzheimer's disease is a degenerative disorder. The proteins, the pathology in the brain are probably building up over many, many years and decades. So a current strategy now in the field is to try to define identifiers of that disease process very early on. So with the cardiovascular analogy, if you're 45 and you have an elevated cholesterol, most people would recommend we intervene at that now, if not even earlier, with diet, exercise, medications if necessary. That's where we need to go in Alzheimer's disease, and that's where we're going.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
Alzheimer's disease is a complicated disease. We don't know if you like, the order in which things go wrong in Alzheimer's disease. And genetics allows us to get to the beginning of the disease. It allows us to say, what comes first? What mutations, what variants do we have right from birth which can predispose to disease? The great thing about genetics, in a way, is it's a completely unbiased way of looking at disease causation, and that's why it's been so powerful.
The mutation in a gene changes the gene very slightly. That changes the information that that gene uses to make a protein. So the protein that a gene with a mutation makes is just slightly different than the protein that a gene without a mutation makes. And that small difference, and it's typically one amino acid change out of hundreds, causes the disease.
In Alzheimer's disease, we see some families who have a mutation, a very simple inheritance of disease. In those families, children who are born in such families have a 50-50 chance of getting disease, and the disease onset is early. More typical Alzheimer's disease occurs once or maybe twice in a family and shows a moderate amount of familial clustering. In the 80s and 90s, we started to look at families with hereditary Alzheimer's disease. I got a letter from a woman whose father had early onset Alzheimer's disease, and 4 of his 9 siblings also had it. We went through all of the genome in all of the individuals, looking for parts of the genome which were only inherited by the affected individuals. And what we found after about 4 years' work was that all of them shared the same copy. All the affected members shared the amyloid gene, shared the same copy of their amyloid gene. And when we sequenced that amyloid gene, we found a mutation, a change in the gene, a mistake in it, which we then realized caused the disease. All of these genetic causes related directly to the amyloid that was built up in the plaque. All of the mutations had the same outcome, and that was that more amyloid was produced by the metabolism of these genes. And so that really led us to believe amyloid was at the center of the disease process.
Since we know that the protein in those plaques is important, it's important to target drug therapies or any kind of intervention. It's important to say, let's get rid of that plaque. Let's reduce the amount of protein that ends up in that plaque. And that genetics provides the rationale for spending, the hundreds of millions and billions of dollars to develop drugs. You don't want to be working on something that's not related to the disease. You want to be working on something that really causes the disease. We had autopsies from both the early and the late. And you compare that early onset Alzheimer's to the late onset Alzheimer's disease, and you really see the same. You see the plaques, you see the tangles, you see the distribution, you see the shrinkage of certain parts of the brain, you see all of the same changes. even though the genetics was night and day different. The early onset families, we knew you could look at the pedigrees, look at the family structure, look at the family tree, and say there's one gene that's causing Alzheimer's in that early onset family. Late onset families, we couldn't say that. It was much more ambiguous, much more puzzling.
What predisposes in the typical late onset Alzheimer case who gets disease in the 70s and 80s with a very unclear family history? In those people, we know it isn't a simple mutation. We know that. We expect it to be a mixture of causes, a mixture of predispositions, some environmental and some genetic.
We think that there are a lot of different genes that make very small contributions to whether you're going to get the disease or not. We call these susceptibility factors. No one single susceptibility gene makes that big of a difference whether you're going to get it or not. But if you have 5 or 10 susceptibility genes and you've got the bad form of all 5 or 10, that's going to increase your risk. Maybe it's 10, maybe it's 20, maybe it's 30. Maybe there are 30 or 40 susceptibility genes that we need to identify, any one of which makes a very small difference about whether you get the disease or not, but you put enough of them together and that increases your risk substantially. Genome-wide association studies are designed to give us many more susceptibility genes. This technology that we're using is just phenomenal, and it's really caused genetics of late onset to really explode in the last few years. The DNA is put on these very tiny chips that have 500,000 to 1 million spots that looks at 500 to 1 million sites in that person's DNA. And each one of those we have to look at and say, is this a good drug target? Is this the one we want to really focus a lot of money and attention on? And the more susceptibility genes you have, the more candidates for drug targets you have, and the better chance you'll find one that this is a good one, we're going to aim at it, and we're going to put a lot of money into it and see if we can prevent the disease. We do have one well-defined susceptibility gene called ApoE, or apolipoprotein E. It's genetically variable, so you can have different forms of ApoE, just like you can have different eye colors. It's genetically determined. One of the forms puts you at high risk for heart disease. That same form puts you at high risk for elevated cholesterol, and that same form puts you at elevated risk for Alzheimer's disease. And in fact, the risk, its effect on risk for Alzheimer's is probably the strongest. Number one, what that tells you is that it's the model, it's the poster child for the susceptibility gene concept. It's a gene we all have. You can grow old with the bad form and not get Alzheimer's, although your risk is increased. So it doesn't absolutely determine that you'll get Alzheimer's, but it changes your risk. As people get old, everybody's risk gets pretty high. So one thing is, if your parent had Alzheimer's in their early 80s, like my dad did, that's probably not that atypical. So somebody getting Alzheimer's that late, as a geneticist, I think maybe he didn't have that many susceptibility genes, therefore I probably might not have that many susceptibility genes. So I'm not really that worried about my risk.
It's really an exciting time to be doing genetics because we can do things now that we couldn't dream of doing 10 years ago. We can do these surveys of across the genome in thousands of individuals relatively quickly now. We couldn't have dreamed of doing that 5 years ago. We just couldn't have dreamed of doing it. Furthermore, though, in 5 years' time, we'll be able to do far more. We'll be able to sequence thousands of genomes and compare the sequences, the actual DNA sequences of thousands of people with Alzheimer's disease and compare it to thousands of people who don't have Alzheimer's disease. And that will enable us to finally work out what all the genetic predispositions are. It'll be a lot of work and it'll involve a lot of complex statistical calculations, but we know, we're confident, we know we're going to be able to do it, and that's incredibly exciting.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
During the year that I had applied to medical school and was waiting to get in, my grandfather developed a lot of memory problems. And it was really striking because here was a man who'd always been the intellectual leader of our family, who'd always been able to teach me and teach my father, and I thought was kind of probably from whom I'd inherited what brain I had. By the end of that year, he didn't know who I was. He knew that I was someone that he'd seen before, but he didn't remember my name or remember me. And that was at the time I was deciding about going into medicine. I knew already I was interested in the brain, But to see the disease rob somebody of who they were that way was really striking. And as much as I try to stay detached from my patients, when I see that in patients and remember what it was like for me, I don't think you ever get over that. One of the things I think is most important to do as a clinician is to reassure patients and their families that they have a real brain disease, that they're not just going crazy, that they're not stupid, that this is a real brain disease, that there's a disease that's attacking their nerve cells and makes these symptoms occur. So there, I actually think in Alzheimer's disease, it's important to not make a distinction between the mind and the brain, because the mind goes in Alzheimer's disease, but it's because of a brain disease. So we actually have a large number of imaging techniques that we can now apply to the study of Alzheimer's disease. And I think we're going to find that these are really complementary. So we can look not only at the structure of the brain, but also the function of the brain and even the pathology of the brain in Alzheimer's disease. So the imaging technique that people have used most widely is called magnetic resonance imaging. And this is the same kind of MRI that someone comes in and they get for their knee or their back if they've got a spine problem. And you can use MRI, magnetic resonance imaging, to get very exquisite pictures of the anatomy of the brain. And this allows us to rule out things like stroke or tumor, which can actually cause cognitive impairment, but also allows us to look for evidence of brain shrinkage and look for a pattern of shrinkage, or what we call atrophy, in parts of the brain that we know are affected by Alzheimer's disease, such as the hippocampus here, as well as shrinkage in parts of the cortex that are important for cognitive function. Another imaging technique that we work with in research is functional MRI, which is my favorite, and this is where we actually look at the function of the brain during a task. We're stressing the brain, asking it to do exactly the kinds of memory tasks that it has trouble with in Alzheimer's disease. And I like to use the analogy from cardiac disease. So if you want to look at the size of the heart, you can do a chest x-ray, but that's not very predictive of how well the heart functions. If you want to see if they're at risk for having a heart attack, you put them on a treadmill and you stress the system and see if you can induce changes that tell you that that system is at risk for having ischemic heart disease. And I think that we might be able to use functional MRI in that same way. So we can stress the networks that we can see are abnormal at later stages very early on and see evidence of dysfunction before you lose tissue and have atrophy and loss of these nerve cells that will never be able to recover. This allows us to understand how it is that a person's able to form a new memory that will last a lifetime. We found that the brain is actually a very complex set of networks. This is the frontal cortex, an area that's important for attention, for what we call executive function, and focusing you to say, this is important to remember. The back part of the brain, the occipital cortex, is important for vision. So when you're learning a new memory with visual input, that's required to turn on. And that's actually where people recognize faces. And when you recognize a face of someone you've seen or know well, that's the area of the brain that's important for remembering a face and then linking it to the other information. And that's where the hippocampus comes in. Buried deep here in the middle of the brain, it forms these links. And the brain is amazing with all of these different areas. What the hippocampus can do is integrate that input into a cohesive memory. So you not only remember a face, you remember their name, the context in which you see them, what music was playing, what it smelled like, a very rich and complicated memory. We've learned that the coordination between the hippocampus and the precuneus is actually critical in forming a new memory. And when you retrieve a memory 30 minutes later, these are the areas, particularly the precuneus, that are going to turn on. And we think that these areas of the brain are very important for knowing that you know. So not just retrieving the memory, but having confidence that the memory is correct. One of the striking things that we've noticed is that where amyloid, this abnormal protein that accumulates in Alzheimer's disease, is depositing in the brain, particularly in the precuneus and out here in the lateral parietal, are really strikingly overlapping to those areas that need to work during memory formation. And we think it's not an accident that that's actually where amyloid is forming early in the brain and why memory might be such a vulnerable cognitive function early in Alzheimer's disease. One of the most surprising things we found is that some parts of the brain actually hyper turn on or hyperactivate. That was really surprising because the original idea was that as progression of Alzheimer's disease that you just lose everything and everything goes down in a nice linear fashion. But it actually turns out that some parts of the brain, particularly early in the disease, that these parts of the brain, the hippocampus that's so important for memory, goes into hyperdrive. And in order to form a new memory, it actually works harder. And we've thought that perhaps this is evidence of compensation, or trying to compensate for early Alzheimer's pathology. So it's revving and driving as fast as it can, and during that phase of hyperactivity, you're able to maintain memory. And unfortunately, what we've seen is that as people progress, they lose activation in exactly the area that was hyperactivating. So although this may be compensatory early on, it's also a sign of impending failure of the hippocampus. And more we've seen that this hyperactivation is actually a very good predictor of who's going to decline over the next 2 years. What we really need in Alzheimer's disease is to make a link between the pathology of Alzheimer's disease, such as amyloid, and the later disease. And we need to do that at a point where you could still prevent the disease from progressing. There are a variety of clinical trials now trying to either decrease the production of amyloid or clear it out of the brain using immunotherapy. These are types of vaccines or using antibodies to try to clear the amyloid out of the brain. And we've had the opportunity in these studies to study a small number of individuals before and after they get antibody treatment. And this is actually a case of a 76-year-old woman who had very mild Alzheimer's disease when she started the study, but had already really lost hippocampal activation. And in this first study, we looked at a single infusion of antibody, and were able to scan her 12 weeks after this first infusion. And we're able to see that, in fact, not only did she get better clinically, but we saw evidence that we could restore hippocampal function. Now, this was from a very small study, and it's difficult to draw conclusions from single cases. I do believe that we've got the cure for Alzheimer's disease in someone's test tube. I do. I think the question is, how fast can we get that into clinical trials, and do we have the right drugs in clinical trials? And actually, the thing that keeps me up at night, the thing that I'm most worried about, is that we're testing these drugs too late in the disease. That once a person already has mild dementia or is at a stage where they've already lost their hippocampal activation, the chances that we can rescue that brain, even if we've got a terrific drug, and prevent further decline are very small. I think we're going to have a better chance as we move back earlier in the disease to mild cognitive impairment and ultimately perhaps normal people who have the earliest changes of Alzheimer's disease in their brain.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
The demographic transition that we are experiencing in this country and worldwide, the number of individuals we are going to see at ages which have put them at high risk for Alzheimer's disease, coupled with the scientific opportunities we now see, makes this a time at which it is critical for the public to understand both the challenges and the opportunities and to become a part of addressing the solution to Alzheimer's disease. Through all of the history up till present, there has been a far greater number of young children aged 5 and under than older people. And I think we can say with some certainty this has been true since the evolution of the species. What is being shown in data analyzed here is for the first time an enormous, a dramatic, and a very rapid change in that profile. As the percentage of younger children has decreased and the percentage of older has increased. And by the year 2050, we'll reach a point at which there'll be some 2 to 3-fold more people aged 65 and older than aged 5 and under. A dramatic change in the profile of the human population. For the 100-plus group, which actually will become a significant, increasingly significant part of our population, there will be a 4 to 5-fold increase, a 400 to 500% increase in the number of individuals 100 and older between 2005 and 2030. Age is a critical risk factor for Alzheimer's disease, the risk doubling with each 5 years over age 65. Therefore, a large part of understanding the challenge that Alzheimer's disease presents to the future, even more than to the present, comes with understanding what is happening to the age profile of the American population. And one can see, for example, in the chart plotted here, which show the number of individuals from the year 1900 out through projected 2050 who are age 65 and older. And the year 2000, for example, are estimated to be some 35 million individuals over age 65. By the year 2050, this will increase to some 87 million, more than doubled. The upper parts of each of these bars, shown in red, is the portion of that over 65 population that is aged 85 and older, the population at greatest risk for developing Alzheimer's disease. The human costs of Alzheimer's disease are enormous. The affliction, the pain for individuals with disease, and at least equally so for family and loved ones. And it is above all this, I think, which motivates our efforts toward research. In addition to these human costs, however, there clearly are financial and societal costs which are also substantial. Estimates vary, but those estimates, which include both the direct costs of care, the costs of caregiving, the loss of productivity, generally exceed well over $100 billion per year in the country now, and are projected to increase as the projected number of individuals at risk for disease increases. If we fail to cure or prevent Alzheimer's disease in the years and decades to come, we are going to see a several-fold increase in the number of individuals with Alzheimer's disease at cost of human suffering and societal and financial impact that is frankly hard to imagine and hard to imagine sustaining. In this context, the investment that we are currently making in Alzheimer's disease research will not only bear critical fruit in terms of preventing the human suffering that afflicts those with the disease, but as well in providing a financial dividend manyfold the investment in what will be saved in the costs of caring for those with Alzheimer's disease. Our intensive research efforts focuses on decreasing the risk of Alzheimer's disease by treating and ultimately preventing disease. This is a time when the scientific opportunities are enormously exciting, unprecedented. We are now able, as we could never have imagined before, to actually search all the genes in the genome for any of those genes which might influence the process of Alzheimer's disease. We've been able to, by transferring human genes into mice, actually reproduce characteristics of the disease, to test new treatments. The imaging of the brain that we're currently able to do could not have been imagined 20 or 30 years ago. There's enormous interest now in what the effects of lifestyle interventions such as exercise and diet may have on the risk for Alzheimer's disease. Science does progress through a succession of discoveries. There's a necessity to have increasing increments of knowledge, and this does, unfortunately, limit the speed at which we make progress. And it pains all of us that that is the case. It pains the researchers working in this area, as well as those who see the ravages of the disease. Our commitment, therefore, is to turn from the disappointment we feel at not yet having a cure to a conviction and a commitment to pursue an answer as rapidly, as expeditiously as we can, so that we minimize the probability that a few years from now, someone else is going to turn and ask us the very same question: why is it we still don't have the answer?
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
In the body, insulin has a very important role to play in a number of different systems. But for a long time, it was unknown whether insulin had a role to play in the brain as well. And work over the last 20 years has shown that, yes, it does. It plays some very important roles in brain function. Insulin has a very important role to play in aging of the body as well as aging of the brain. In fact, one of the most important predictors of how successfully you'll age is how intact your insulin system remains as you age. So insulin is produced every time you eat. It's a peptide, which is a small protein, and it's produced in the pancreas in response to glucose. Glucose is the primary fuel for all cells in the body as well as in the brain. But it needs insulin in order to be able to be transported into the cell to do its work. If insulin cannot allow glucose to get into neurons, then they are not able to carry out their functions relating to thinking and memory and attention effectively. When insulin is not working correctly and glucose builds up outside the cell, that process initiates insulin resistance. Over time, it may reach a level that would cause a person to be diagnosed with diabetes. And diabetes is defined as having glucose levels exceeding a certain level. Even if a person is not diagnosed with diabetes, but manages to keep their glucose levels below the threshold for diabetes, this is still not a benign condition. Insulin resistance is still happening. The insulin levels are very high. The cells are not getting the glucose that they need to function properly. So this can be a very malevolent condition, and over time could cause increases in inflammation, beta amyloid accumulation, and a number of other negative consequences. Insulin resistance is an underlying pathology for almost all vascular risk factors. So insulin resistance leads directly to cardiovascular disease. It leads to hypertension. It leads to small vessel strokes. And it leads to diabetes. So it is the unifying underlying pathology for all of those risk factors, each of which as we know, contributes to the risk of Alzheimer's disease. The estimates would be somewhere in the order of 25 to 30% of all adults over 60 years of age have insulin resistance. So it's a highly prevalent condition, and it's a condition that's becoming epidemic. with the lack of activity and changes in our diet. Insulin resistance is beginning earlier in life than we might appreciate. We believe it starts in midlife, and this is a period in which many people begin to experience obesity and body weight gain and other metabolic changes. And it's the time at which insulin resistance and high levels of insulin also become apparent. Simultaneously, we believe beta amyloid is increasing in large part because of these changes in insulin resistance and insulin. And as a result, we see symptoms begin to occur, problems with memory, which worsen over time until a person might be on their way to developing Alzheimer's disease. I think insulin resistance is a particular health concern because most adults are unaware that they have insulin resistance. They may have insulin levels that are 300 times normal levels and still be completely unaware that they have this condition because, in part, their glucose levels do not exceed the threshold for diabetes. They're told by their practitioners that they're fine. So the awareness that one should have their insulin resistance assessed as well as their glucose levels assessed, I think, is a very important message. You can have your insulin level tested while you're fasting, and that's somewhat predictive. You can have it tested after the sugar challenge, a procedure known as an oral glucose tolerance test, and that will be very informative. We're hampered a bit because there are no standard assays for insulin, so a test that you would get in your doctor's office might not be comparable to a test I would get in my doctor's office. And work is underway to correct that, to make an easier, more standardized assay. But I think it is an underappreciated health risk. Insulin resistance is in most cases eminently treatable, and there are a number of ways to treat it. By far, the most potent treatment of insulin resistance is exercise. Aerobic exercise improves insulin function dramatically and immediately. So one bout of aerobic exercise, a 30-minute aerobic exercise period, will improve your insulin function for 24 hours. And then diet certainly plays a role. So a diet that is rich in good fats and complex carbohydrates and low in bad fats and simple sugars offers some benefit. And interestingly, doing both of those things together seems to offer a synergistic benefit. We're understanding that fat is not fat, that there's good fat and bad fat, and that whether or not fat occurs in different parts of the body has a different effect. So that fat that is located around the organs, the so-called visceral fat, acts like a gland. It secretes hormones. It is not inert. It's not an inert substance. And that fat depot is responsible for inducing insulin resistance. The fat that lies just under the skin, the subcutaneous fat, is actually beneficial, and it works to take up triglycerides and other bad fat from your bloodstream. So it's not good to have too little fat. You need some fat to function effectively, and the subcutaneous fat is the beneficial fat, and the visceral fat seems to be the bad actor, seems to be the fat that's causing inflammation and insulin resistance. When you consume a meal that's very high in saturated fat and high in sugar, your insulin levels skyrocket. And they remain elevated for a very long period of time. And this is a very abnormal state of affairs. Insulin is designed to rise quickly after a meal and then be cleared very quickly as well, and that's how it works best. But the saturated fat impairs the clearance of insulin, and the high sugar causes the insulin levels to spike. So as a result, you store more fat after you eat this type of meal, and your body is subjected to very high levels of insulin for an abnormally long period of time. In this study, we asked participants to consume a high-fat, high-sugar diet for a 4-week period, or a low-fat, low-sugar diet for the same amount of time. On the left here, I have foods from the low saturated fat condition: oatmeal, soup and lean meats, chicken breast and vegetables. In the high saturated fat condition, we have bagels and cream cheese, we have Philly cheesesteak, we have meatloaf and gravy and mashed potatoes and biscuits. All foods high in saturated fats, and all foods that are commonly found on the tables of Americans every day. The participants in the high-fat diet showed increased beta amyloid in their spinal fluid. And the participants in the low-fat diet improved their profile. They had lower insulin levels and lower levels of beta amyloid. So even 4 weeks of eating well produced a benefit for these individuals in terms of their insulin resistance and in terms of their amyloid profile. This is the amount of fat that is taken in by many people who eat a typical Western diet. And it's rather hard to believe when you see it laid out in front of you. The equivalent of 42 sticks of butter were consumed over the 4-week period by participants in the high saturated fat condition. The message is that it is very important to pay attention to the type of fat one takes in. And we actually know that lowering saturated fat and consuming good fats like olive oil, which is shown here, has a beneficial effect on insulin resistance. One of the functions that insulin plays in the brain, we believe, is that it affects memory. It does this in a number of ways. It does it through promoting glucose uptake in the hippocampus, the area of the brain that's very important for memory, and it does it through strengthening the connections between nerve cells, which is thought to underlie the formation of memories. A memory is simply a pattern of connections between neurons. That's all a memory is in the brain. And so anything that affects or modulates the connections of synapses is going to play an important role in memory. In an abnormal situation such as insulin resistance, there may be some compromise in memory. And in fact, studies have borne this out, that older adults with diabetes and older adults with insulin resistance, in general, have worse memory performance than healthy seniors. One of the exciting things is that we've been able to take these early studies that demonstrate some of the mechanisms through which insulin resistance might promote the risk of Alzheimer's disease into the therapeutic arena. And in particular, one of the studies that we're very excited about is a study in which the goal is to correct the deficiency in insulin in the brain that occurs with insulin resistance. The way that we've found to accomplish that goal is through administering insulin intranasally.
is. So just put it up to your nose and get comfortable. You're going to breathe in through your nose, out through your mouth. Start.
When insulin is administered through the nose, it travels to the brain very rapidly and can have effects in the brain. It does not affect insulin levels in the body. So it's a very effective way of getting the insulin exactly to where it's needed. And the way that we think insulin gets into the brain is that it follows two pathways, one of which goes to the front part of the brain, or the frontal lobes, and the second of which goes more medially and reaches areas like the hippocampus. We know that the hippocampus is very rich with insulin receptors, and both the frontal lobe and the hippocampus are sites that are affected very early on in Alzheimer's disease.
My memory started changing when I realized that I was making mistakes driving my grandson to and from school. And it only took about 3 or 4 times to realize that there was something drastically going wrong. So, you know, I put it off for so long, and then I came to the thought that, well, maybe it's just an aging process. Well, I was wrong. And as it progressed, I couldn't deal with it. It was just, now what do I do? You know, I can't do this, I can't do that, you know, and I don't want to endanger anybody.
If we were in noisy places, he would just go to another room. If we were at a party, he would go sit in another room. Just didn't want to engage.
The feeling is quite terrifying because you don't know when it's going to end. You know, you have the feeling that I'm going to be responsible. I can't do anything about it.
One of the reasons we're very excited about the intranasal insulin studies is because of some data that we've just recently analyzed. So you can see in this patient before beginning the study these prominent areas of low metabolism. So red is the lowest metabolism of all. And then after 4 months of treatment, if you compare these directly, you can see there are fewer abnormal areas. And you can see from this view as well. It looks as if the intranasal insulin treatment improved glucose metabolism for this patient. And this is one of the key mechanisms we think that might underlie improvement in memory and attention with intranasal insulin treatment.
The sniffing test was good because it made my head feel clear.
The differences were subtle, but they were cumulative. They were there, and there was no denying it.
I would back away from conversations because, you know, I couldn't keep up with people, you know. But now, I mean, I go to one of these little party type things, you know, and believe it or not, I gave them my whole life story. I wanted everybody just, really? Is that Jose? You know?
I'm with my husband again, whereas before it was, I don't want to say somebody that needed to be taken care of, but there wasn't any give and take as far as the conversation was concerned. It changed our whole outlook.
One of the most exciting things about our work, I think, is the prospect that it offers for preventing or delaying dementia. And I think the idea that you can delay the onset of Alzheimer's disease is a very important one. Estimates are that if you delay the onset just 5 years, you'll reduce half of all the cases. So by preventing insulin resistance, you may, in the best case, prevent Alzheimer's disease from developing at all, or I think even more modestly, you could delay the onset of the disease.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
Inflammation is a very complex set of body processes that are geared to identifying, attacking, and removing foreign invaders and objects from your body, abnormal materials in your body. So having inflammation get loose in your brain is a very major concern to every neurologist and neurosurgeon. Inflammation in the Alzheimer's brain essentially begins with what you see right here. This is a picture taken through a microscope that's very, very like what Alois Alzheimer saw back in 1906 when he looked in his microscope at little sections of the brain of one of his demented patients, Auguste D. And he used a stain very similar to this one, and he saw black freckle-like things all over the thinking parts of the brain. When he looked at areas of the brain that were involved in movement, he didn't see them. But in the higher centers of the brain, he saw millions of these black freckle-like things. We now know that these are made up of a molecule called amyloid beta peptide. They're highly, highly insoluble. And so when I first looked at this, I said, gosh, it just looks like somebody took some mud or dirt and just threw it at the cortex and hit it everywhere. Surely that would stimulate inflammation. But the dogma was that you couldn't have inflammation in the brain. It was immunologically privileged. And so I went ahead and looked anyway. And I got a chemical that stains things that are involved in active inflammatory response. And that's exactly what I saw. I saw thousands and thousands of little wiggly things, and I didn't know what they were. What I found really interesting, though, was that these cells that were resident in the brain in an Alzheimer's patient lit up with this marker that said, I'm involved in an inflammatory response. And that was big news. What is inflammation about? Why does your body have it? Well, your body uses inflammation to rid itself of foreign invaders and abnormal molecules and toxins, things like bacteria and viruses. Those stimulate inflammation when they're present in the brain or anywhere in your body where they shouldn't be, and inflammatory molecules and cells move into that area and attack and cleanse the body of those foreign invaders or foreign material. Amyloid beta peptide is clearly abnormal. It's almost like a splinter in your brain, and you've got thousands of them, and so you're going to have an inflammatory attack as the inflammatory cells move in and try to get that out of the brain. We stain the amyloid here with a brown stain, and look in the bellies of the microglia. They're full of the amyloid. They actively eat this stuff up, and that's what you would expect in an efficient inflammatory response. You attack things, and you clear them and remove them from the body. Obviously, in the Alzheimer's patient, this may be working, but it's not working well enough because the patients have thousands and thousands of microscopic amyloid deposits. But we suspected that this process was ongoing. It was just a tough assignment for the microglia because, for one thing, amyloid is so very insoluble. Inflammation is always a two-headed sword. It destroys, and that's beneficial. When you have foreign invaders, inflammation destroys those foreign invaders. The problem is it can also destroy healthy tissue in the process, so you've got to be very careful about that. Let me give you an example. When you cut your finger, you may get some dirt and bacterium in the cut, and the cut then becomes inflamed. You have inflammation as inflammatory molecules and cells move in to cleanse the wound. And that's how inflammation does it. It's not magic. Inflammation just destroys everything in sight. So the dirt and the bacteria, they're attacked and removed. And if there are any healthy cells in the way, they get attacked and removed at the same time. Now, this bad side effect in most of your body isn't such a big deal. You grow new skin cells and muscle cells every day. So you can quickly put back the innocent bystander healthy cells that got killed by inflammation in the process of cleansing this wound. Over time, this is going to, we believe, kill a lot of brain cells and particularly a lot of the processes, the nerve fibers that connect nerve cells over time. It will take some time because it's a low-grade inflammation, but we think that it is one of the major causes of damage in Alzheimer's disease. Our tissue bank gets brain samples so quickly after death that we are able to harvest living cells. After you die, a number of different cell types in your brain, microglia in particular, will live on for a couple of hours. And we have developed techniques to get them out of the brains of patients who've recently died and keep them alive in a test tube. And we can use them then to find out what the cells in the Alzheimer's brain were doing.
We're just looking at one piece of the brain. And there's probably usually about 14 or 15 slabs. And this is just one slab. It's right frontal cortex.
The story here is that we can use these cells as a living target. If you get the flask and sure. Show us, I think, live, that would be good. So what do we got here?
Here we got happy nerve cells. A lot of connections.
You're right. I mean, that's one of the things that we know that nerve cells like to do is make connections with each other, and they do that when they're healthy and happy. Okay, so what if we put these same nerve cells together with microglia? And what if we add amyloid to this mix? What do we see? Ah, wow. So here, the microglial cells have puffed up. They're much bigger. They're filling with little vessels that contain digestive juices, so to speak. The most important thing here is the connections between the nerve cells that were so vivid in the previous image. They're gone. They're just gone. And this parallels very closely, actually, what is seen in the Alzheimer's brain. When we combine those two, microglia, and we let the amyloid activate them into an inflammatory state, make them angry, we get damage to nerve cells. Dale Schenck at Elan Pharmaceuticals has looked at one side of these processes, the beneficial side. He said, well, Joe has shown us, and we have confirmed that microglia can remove amyloid and potentially amyloid deposits in the Alzheimer's brain, but they're not doing it well enough. Obviously, they're not doing a good enough job because the amyloid deposits, there's still ever more of them developing in the Alzheimer's brain, and obviously the microglia haven't been able to get rid of them as they should. So maybe we should give them some encouragement and some technical help. And what Dale came up with is, I think, one of the most creative and brilliant ideas that we've had in Alzheimer's research. And that was, he said, let's immunize people against their own amyloid.
So there's a variety of approaches to try and block the progression of Alzheimer's disease. Once you have it, how can you go after the plaques or the tangles, try and reduce them, remove them, so that you can change the course of the disease? And there's a lot of sort of logical ways to do this, and there's some surprising ways to do it. One of the logical ways to do it is to reduce the production of beta amyloid from the get-go. The more surprising way to do it that we discovered in the late 90s is that if you actually immunize or vaccinate with the beta amyloid peptide itself and stimulate the immune system, that the immune system can then actually either totally prevent, or reverse the plaques in the brain tissue. Back in the late 80s, a group of people came together and felt that it was really important to work on Alzheimer's disease. And we had a few ideas about how to do it, but we really didn't have specific tools in terms of how to do it.
We needed the complexity of a living system. As much as we'd love to be able to just model it in a computer or, in a dish, we just can't. There's so much about the brain that we still don't understand to this day, but particularly back then, we really were in desperate need of a testing system, and we tried many, many things.
It wasn't until the early 90s that we finally had a decent animal model. And basically what that was is we took a gene which was rare but known to cause Alzheimer's disease and put it into a mouse.
What a transgenic animal is you introduce a snippet of a human gene or a bigger piece of the human gene directly into the genes or the genetic makeup of a mouse. But the added piece is that this disease takes 10, 20, maybe even longer, 30 years in a human, and we can't wait 30 years to begin an experiment. So we had to take a chronic disease, speed it up, and make it happen in a matter of months in a mouse.
The moment that we had that was in the mid-90s, we all sat down and said, oh my God, we have a model now to test a lot of the ideas we've always wanted to do. And so we brainstormed. We thought of all these different things we could do with this mouse. And the idea was to vaccinate. Just like you get vaccinated for polio, vaccinate with a synthetic piece, a little peptide protein fragment of the beta amyloid into the mice. And when you get vaccinated, what do? You raise antibodies in your blood. And that's what protects you if you're being vaccinated against some infection. Here it's a little different. What we hoped is that those antibodies would circulate through the body, and what antibodies do is they bind to whatever they've been made to. And we hope these antibodies would bind to the beta amyloid and help clear it, help get rid of it. Very simple idea, extremely simple idea, but there were 100 reasons why it shouldn't work. Sometimes in science, if you have a very simple idea and you can't actually completely kill it, you should just do it. Because if it works, it works, and then you figure out why, as opposed to not doing things because there's good reasons not to do it. And so we did it. This is what you see in untreated mice. This is the brain. The brown material is the plaques. And after they've been vaccinated, this is what you see instead of that in the treated animals. And it was so black and white and so obvious that I remember Dora saw this first.
We were at the bench looking at through the microscope, and it was a nice time because whenever we would see a plaque, everybody would come running down to see the first plaque ever in an animal model. And then when we didn't see a plaque anymore in the animal model, everyone came running down to the microscope because it was the first time we had seen something, and then it was the first time we could see it go away. And of course Dale very quickly thereafter was commandeering the scope because he
Well, as a biochemist, to dissolve plaques from an Alzheimer brain, you have to take basically the strongest chemicals you have on your laboratory shelf that won't completely destroy the protein, but the strongest dissolving reagents that we know. And that's what it would take to dissolve amyloid out of an Alzheimer brain. And the fact that microglia can do this, under a normal cellular condition without causing apparent harm is just truly amazing. The field moved incredibly fast.
We thought that the antibodies that the person or the mouse model would make, the mice would make, would do everything. These antibodies would bind to the beta amyloid and help clear it, and that would be that. What we had no idea about was that what we were really doing was doing that, and we were triggering or facilitating the ability of the immune system to now go after the plaque with full force. And once the antibody binds to these plaques in the brain, these microglial cells come along, see this antibody sitting there, and then it triggers a whole response where they literally engulf and eat. They're called phagocytes for eating. They engulf and eat and digest the plaques away. And if you look on here, as you go forward, in green is one such microglial cell doing exactly that, caught in the act.
So let's take a look at more resolution of what's happening in this site here. We have a beta in red. Surrounding that plaque, what is remaining of a plaque, we have a group of microglia cells that are very, very close contact. And you see that the cells touch the surface of that amyloid plaque that now is very smooth, indicating that the cells digested the surface of that plaque that was very fibrillar in the beginning, and what's remaining is the surface of what probably, if we wait longer, will disappear eventually. So that indicates that those cells are internalizing the A-beta with the help of the antibody and digesting the A-beta.
Once we realized that we could actually treat our mouse model of Alzheimer's disease with the beta amyloid peptide, in other words, by vaccinating them or immunizing them with the beta amyloid peptide, we immediately realized that that had potential for treatment of Alzheimer's patients.
Earl was having trouble at work. And so they were trying to figure out exactly where to place him so that he would be most effective. And I think that was those started to be signals of things that were actually not going very well. Let me pick up your foot. Mm-hmm. And bring it over. There you go. Mm-hmm.
Something was wrong. And I just said, it'll come around. And I kept seeing that it wasn't going away. And then I started getting antsy. I thought, what the hell is going on?
I remember Earl reading 5 books at a time. I would complain because he had books all over the bed and all along the bed. Yeah. But he read 5 books at a time. Like I say, worked on the computers, wrote in Fortran, did all these things which he was giving up.
I thought it was the end of me, because I knew about Alzheimer's, but I never thought I'd get it.
It was interesting how we got involved in the Elan trials, the 2001 Elan trials, because our son-in-law saw it advertised in the paper, and he called us and said, I think this is maybe something you'd be interested in.
Mm-hmm. Yeah.
and we were interested in it. So Earl and I ran out there to get involved in it. And of course, we think there should have been a lot more people that were anxious, but we were one of few that were involved, I think, and excited about it.
I was just hoping that something would surface that would help me.
We began what we call clinical trials, and that's a very long process, actually. And the way it's done is most of what you do is make sure that what you're doing is safe. So you do a few number of patients, very controlled environment, very careful amounts, etc., and show that it's safe. And we call that phase 1 trial. So we did that, and everything looked fine. And then we went on to large studies. And now we're talking several hundred patients, 370 patients, where we did what's called a phase 2 study, where, again, a lot of it is about safety. Most of it's about safety, but you're starting to look at things like, gee, is this working? Are the patients performing functions better? How are they doing?
He had 3 injections, probably over a period of 3 months or something like that, maybe even longer. When he was given the first injection, he had a bit of a reaction to like a protein, an allergen type reaction. And I said, I think you got the vaccine. And sure enough, he did. And so it was exciting because we were actually feeling like it was doing some good, even initially.
It gave me a lot of hope, and knowing that it was going on.
We were just well underway, everything was going fine, and after the second dose, we ran into a problem with encephalitis in a very small subset of patients, about 5 or 6% of them. And although it was transient, it was severe enough that we felt it was prudent to stop dosing.
When they ended the trials, well, we were cussing. We actually were angry. We said, I kept saying, oh, that was a mosquito in France. It doesn't have anything to do with this vaccine. But I think back and I kept saying to everyone who asked, so how's Earl doing? I would say, he's maintaining. He's maintaining. And that was really important because you were functioning well at that time. And just to stay there would have been a blessing because it was working. So, and we said that for many, many years. I'm thinking back now, we probably 3 or 4 or 5 years said, he's maintaining. Everything is good. Just recently, I would say we have seen a shift.
To our amazement, if you look at the bottom row here and compare it with the top, at the top is the typical sort of pathology that one sees in patients who have Alzheimer's disease. The top 3 are brain sections where the brown is the beta amyloid, the typical plaque-like appearance. On the bottom is the comparison of what you see after a patient has been vaccinated, where there's brain regions that are nearly completely void. The plaques are gone. We are really very excited that we can literally remove amyloid plaques from patients suffering from Alzheimer's disease. And as we move forward now, what this means is since we can do it, we're developing new treatments, things like bapineuzumab, which is a very long word, fancy word for a human antibody that we make in the laboratory, in a very large laboratory, that is moving through the process of clinical trial.
Does that feel the same when I touch you on both sides? Feel the same?
Same.
Yeah.
Instead of the patient having to make the antibodies and do the work, we're providing the patient directly with the antibodies. Now, the good part about doing this is that you can give exactly the amount and the dose that you want to give. In other words, it's much more controlled when you give the antibodies, and so that's what bapineuzumab is. And so that antibody circulates around, and a little bit of it, this bapineuzumab, gets through into the brain, and if there's plaques there, it will bind to those plaques and neutralize them or stimulate the clearance of them.
This is one of the leading drugs that's in the horse race to get an approval. The sponsors actually have moved this drug into phase 3 because they are very excited about its progress. Dr. Gargas was a surgeon in Wisconsin for many, many years.
Well, yeah. Well, I think I'm very, I walked into something that was very good. And I think that I'm getting real good treatment.
I think one of the few things that experience can teach you is you actually get a sense for when the science is very rich and it's a magical time. And it doesn't happen very often, and I think the field is at that point. The diagnostic tools, the understanding of the disease at so many levels. Actually, perhaps the most important thing is that the biology and the clinical understanding are, as we speak, getting put together at last. It's finally getting put together, and we're going to see real treatments happen as a result of that. It's a magic time. It's a magical time for us, and we really hope the patients are going to benefit from this.
We're going into the house, okay?
I wake up every day, and I think maybe today's the day. Yeah, and that's what keeps me alive. That somebody will come up with a
breakthrough.
A breakthrough. Yeah.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
In terms of breaking through in Alzheimer's disease, not only are we making progress in the medical part, but we're also beginning to realize that behavioral strategies in the absence of medication will actually make a huge difference in terms of the way in which we age. And that's a real revelation, because if you asked somebody 10 years ago, how are you going to fix brain aging or what are you going to do? They would have told you it's a medication. Now we know there's great promise in terms of behavioral interventions that can be adjuncts to medications and good health policy. In Alzheimer's disease, a variety of things happen. Amyloid accumulates, cells degenerate, synapses are lost. But one of the givens is that oxidative damage increases in Alzheimer's disease above and beyond what you see in normal aging. And that's a signature of the disease. And we think, and many people think, that it's a serious problem that needs to be addressed and managed. The brain is probably one of the biggest victims of oxidative damage because it's burning most of the oxygen. It's used to make a lot of the nutrients and cellular energy that we need in order to function. Unfortunately, there's a byproduct, and the byproduct is what you call oxidative damage. With oxygen, there's some inefficiency in the way the cell can use oxygen, and it causes this oxidative damage or injures molecules, proteins, lipids, and nucleic acids. And that oxidative damage causes them to dysfunction. To a cell, oxidative damage basically is equivalent to molecular rust. It basically rusts the cell and causes it to oxidize. It's similar to what you see in a Midwest car with a bunch of salt in the road, and you see this chrome and these fenders falling off. So without some antioxidant protection, you're basically becoming a Midwest car versus a California car or something that's been in the garage forever. So you need to protect the brain against oxidative damage and molecular rust. A natural question is, if we reduce oxidative damage, can we improve cognitive function? So we undertook several years ago a study using aged beagles to determine whether an antioxidant diet would improve their cognitive function and improve their learning ability and their ability to remember. We noticed that the aged dog deposited amyloid, one of the hallmarks of Alzheimer's disease, just naturally. They also have natural cognitive decline, just like a person does. They can also be tested by basically the same tests that are used in humans and in higher order primates. So you've got the analogies between accurate translation in terms of the exact tests, and we've developed many of those and used them in our aged dog studies. And then finally, you can get enough of them to do an actual study, where it's really hard to do some of these rigorous studies, with everybody eating the same thing day in and day out, doing the same type of activity. So in the beagle, we were able to feed them the same thing for 3 years every day, day in and day out. So you control their diet and you can control their activity pattern. Try that in a human, for example. It's not impossible. Nobody is going to eat the same thing for breakfast, lunch, and dinner for 3 years in a row. So you can't do it. You can't study rigorously the effect of diet. Dogs, they were fine with it. Much to our surprise and delight, you can teach an old dog new tricks. And that's what we basically did. We taught an old dog new tricks. We found out that an antioxidant diet improves learning, it improves the ability of an animal to remember things, and it actually rejuvenates the brain to be able to perform tasks that it couldn't perform at the same age were the animals not treated. It's quite dramatic. We'd taken and reversed and rejuvenated the brain to be able to perform something that it couldn't perform. And it wasn't because it was losing function, it actually gained function. And that's a very significant discovery that I'm really excited about still, because if we can put quality back into life through an intervention, that's what we're looking for. We don't want to just prevent decline. We want to bolster function. This needs to be really followed up, and we're very excited about it, and it's very encouraging. I don't know of an equivalent of this in human studies yet, but I'm sure if we start looking and design the right trials, we will have a chance to see that, and we'll look for interventions that could possibly, rejuvenate the aged brain. That's what we're looking for. Fruits and vegetables are very rich in antioxidants, and like blueberries bring a whole series of antioxidants. Strawberries will bring some others into the picture. Broccoli is great. Spinach is just a natural source of antioxidants plus other nutrients. Now, every antioxidant isn't as good as the next one, but what it seems to take from modern research is a combination of antioxidants. You don't just want one, you want multiple ones, and they work as a team to sort of minimize the oxidative damage and to help facilitate the repair mechanisms. So if you go into the health food store, you'll find all kinds of bottles that are labeled with all kinds of miracle claims and everything. And I have to say, we probably don't know the absolute optimal one, but some antioxidant intake is essential. You get most of it from fruits and vegetables and not supplements. One of the ways I think about, sort of the brain preserving itself is what would be the most ideal change that you could induce in the brain that would help it maintain function longer and learn better. And so several years ago, I had this idea that maybe it was exercise. It's sort of self-evident that exercise is supposed to be good for you. We know it's good for the heart, and it's probably good for weight control and things like that, but for brain function, it's not intuitively obvious. And so we needed to put some rigor in terms of the hard science behind it. And so I had this idea that maybe BDNF, which is kind of a nutrient or a fertilizer to neurons, which also makes them smarter, might be naturally increased with exercise. The effect of brain-derived neurotrophic factor, or BDNF, on neurons is just absolutely remarkable because it stimulates them, they're healthier, they connect out more, and it's a growth stimulus. It's basically fertilizer to these cells. The question is, will exercise help improve learning? We take two groups. One is just sort of in their cage and don't have access to a sort of a running gym. And the others get in their running wheels at night and they exercise. in order to test their learning, we basically take the two groups and put them in this Morris water maze. They are put in the tank and they don't like the water, and what they have to do is they have to find a little platform that's sort of below the surface. It's really kind of equivalent to finding your car in a parking lot at an airport when you can't really see it, but you sort of have to learn from the cues around the airport of where you parked.
Sedentary animal still has not located the platform at this point.
All right.
We find consistently that the animals that have exercised are able to learn faster and better than the animals that haven't exercised.
All right.
Yay!
Did it.
We looked at their brain, and lo and behold, BDNF was increased in the part of the brain that is controlling thinking and learning and is vulnerable to Alzheimer's disease. And all I could say to myself, and my students would say, holy camoli, that is so smart. The brain knew something and how to look out for itself that we had no idea about. So it kind of has a self-preservation mechanism that also makes it smarter and to learn better. Certainly in animal models, there's no question about it. Exercise can induce growth factors in the brain. It can help build new neurons into the circuits. It can build synapses, improve learning, and it can basically improve vascular function. Oh my gosh, what drug will do that? Nothing. And, it's simple exercise. I think the fact is that exercise may help delay the onset of Alzheimer's disease, but it's not going to stop it. And, what the recent studies have shown in humans is that the probability that somebody will lose cognitive function in the short term is actually reduced by exercise. And there's been multiple studies out. But there's always the possibility in what you call descriptive science, there's some other reason that goes along with exercise, like most people that exercise actually eat better. They probably take better care of themselves medically. So you try and control those things, and you can certainly control for smoking, drinking behavior and other things, but you never know until you do a randomized study that you've absolutely randomized the population and you have an objective answer. When we began to discover the whole BDNF story, I have to say I wasn't the most active, person that exercised on a regular basis. And so I started thinking about it, and I said, holy smoke, what am I doing? Look, I'm doing this work, and I do believe it, so I better do something about it. And I became convinced that exercise is going to help me function better intellectually and feel better. You got to kind of get yourself into a mindset. Do I believe this or not? And you say, can I afford not to believe it? And so to me, I don't think that the risk is worth it.
All right. Good job.
Yeah.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
As we age, our body changes in many ways, and these are normal aspects of the course of life. Our hair thins, our skin wrinkles, and our brain ages. And we can do quite well with these normal kinds of changes the brain experiences, and all of us do experience these changes. There's no avoiding them. However, there's a certain class of changes that not all of us will experience that are diseases, such as Alzheimer's disease. And while normal aging has subtle changes, and we do see differences from our younger selves, Alzheimer's disease causes devastating changes that attack the brain and the mind. And those are the kinds of changes that lead to our inability to function as we normally would. We don't receive those changes passively. The brain fights back. You see forms of compensation. Certain regions of the brain, which normally in a healthy young adult might be quiet, now start to work hard. And what we think is that the brain has a remarkable reserve capacity.
Your head is going to rest in this pillow right here, so make sure you don't hit it as you lie back.
In this task, we're going to have you retrieve what the words were. What we did in this study is we had both young adults and older individuals do a memory task. And then we captured brain activity when they're retrieving these words. And what you see here are the regions of the brain that are active during that effortful attempt to retrieve from one's past. And in healthy young adults, you see very selective recruitment of brain regions, and that's shown over here in the bright colors. When you look over here at a group of healthy older adults about 75 years of age, what you see is a remarkable increase in the amount of brain activity that's used to do the same task. And we think that might be to compensate. As an analogy to your muscles, if your right arm got weaker, you might start using both arms to carry that heavy box. The brain may do a similar thing. As our normal, very selective regions that are recruited become insufficient to accomplish the task, the brain harnesses all of its available resources and works harder.
Okay. I think that's okay.
Now what's going to happen here is you're going to see words that appear on the computer screen one word at a time. And as always, please keep your head as still as possible. And here we see in a 73-year-old, there's a little bit more atrophy. That is, the brain is shrunk ever so slightly. That's very normal. We see that in everybody. This is a remarkably healthy brain. I found it absolutely fascinating to be able to use our studies of normal and healthy memory to look at those as a foundation for understanding how memory changes with aging. And so over here I'm showing you the two extremes that we see in aging. Here's an individual who's an older adult, completely cognitively normal. You don't see much red pathology, which is the amyloid plaques, in this individual. At the other extreme, here's an individual with Alzheimer's disease, has cognitive impairments, and you see the underlying pathology. But there was a case in the middle that we've just started to explore more. And these are individuals who have Alzheimer's pathology, but are symptom-free. These are older adults walking around, otherwise healthy, cognitively intact, but have the underlying pathology of the disease. There may be resources in the brain reserve, that can be called upon to help deal with the injuries to the brain. One sees this kind of brain reserve that may play a very dramatic role in normal aging. I think it's important to ask, can we harness those kinds of capabilities to stave off some of the symptoms of Alzheimer's disease, at least in the earlier stages?
In the religious order study, our research is focused on what can you do in your life to maintain your cognitive health as best as it can be within whatever constraints you have genetically.
Hi, how are you? Did you just get here?
A few minutes ago.
Oh, okay, okay. So now I have some questions about everyday information. Some questions may be easy, but others may be hard. What is the season?
The season is summer.
And what is the date?
Today's date is July 24th.
Okay. And what is the day of the week?
Today is Thursday.
Now I'm going to show you 10 printed words, and then I'll ask you to recall all 10, or as many as you can.
Chicken, cabin, butter, shore, engine, arm, queen, letter, pole, grass.
Now tell me as many of the words as you can
Letter,
recall.
ticket, pole, engine, butter, arm, shore, engine, cabin, and I think the queen.
Good
I think
job.
I might have said queen. Engine, arm, queen, letter, pole, grass.
I want you to recall as many as you can.
Ticket, arm, butter, pole, shore. Ticket, arm, butter, pole, shore. Pole. Shore. I said shore, didn't I? Ticket, arm. That's it.
What is the year?
1951.
And what is the season?
Right now, a fall.
Okay. Now what is the date?
The date is, I think, 1954.
Okay.
How do you develop a brain that, as the pathology accumulates, you don't lose your memory? So let's assume for the moment that the pathology is accumulating and we're not going to stop it and we're not going to slow it down and we're not going to take it out. All right. I still think that there's ways of doing things that impact your brain in such a way that that pathology can march along at its own pace and you might not experience memory loss.
Good morning. I would like you all to know Catherine, who died in Iowa City 2 days ago, gave her body to science. And so there will be a memorial mass in Iowa City. I will put signs up for drivers and for passengers. I'm in the study.
Are you?
Oh, yeah.
And it helps the research.
Oh, it helps research. Oh, yeah,
That's
sure.
right. That's the
only reason we
important
do
part.
it. That's the reason we do it.
That's true.
We are the Sisters of Charity of the Blessed Virgin Mary. We were trained to be educators. This is part of our mission to continue in the Rush study because that's educating. And that's the last piece that we can really do in our life, to continue our educational mission.
Receive Holy Communion, the body of Christ. There, very good.
There are people upstairs in our Alzheimer's unit right now that are close friends of mine. And it's hard to see these highly educated, intelligent women losing that gift of intellect that they have.
Let's see. Ah.
And then watching them deteriorate physically as well.
Thank you.
So It's not easy, because many of them are my friends.
So these are two brains from participants in the religious order study.
They both have significant atrophy. You can see the atrophy here and here. This one does look more severe to me.
The religious order study involves older nuns, priests, and brothers without dementia who agree to be tested every year to tell us all kinds of things about themselves. And a condition of entry is that they have to agree to organ donation at the time of death. We're interested in what are the common changes in the brain that cause people to lose memory as they get older.
We want to see how we can figure out how to age without manifesting clinical signs of dementia or memory loss. I think this is a good example here of atrophy or shrinkage of the brain. So when you get older, everybody's brain shrinks. Nobody's immune to the shrinkage, just other things that happen when you get old. You get gray hair, your brain loses some substance. Doesn't mean that you're losing cognitive function or getting demented. You actually lose substance. And we look for how much substance is lost in the brain. When there's a severe amount of loss, we are suspicious that there's something going on. More likely in Alzheimer's disease, for instance, does your brain shrink a lot. In this brain, you can see here this huge gap. The brain should really be continuous across here. It's a very common area for atrophy in Alzheimer's disease, but we're going to say how severe we think it is, and then we're going to be able to relate that to their cognitive function. And that looks like, at least for this brain, right here we lose the cortex. From here to here, it just seems to be gone. And that's a stroke. So a very small, old stroke, could have been years that this person had it.
So taking all these different pieces of the puzzle, we can begin to put together how are different risk factors that are causing memory loss, what's the pathway, how are they doing that, what are they doing to the brain to actually cause the memory loss. And so from these large data sets, you can draw inferences about the biology of the disease, the causes of the disease, and how the causes lead to the biology. And it's that interface that's going to lead to strategies for prevention. And that's what the hope is.
We used to be taught that your brain was just something you were given. It was finished developing in your 20s, and then it just was a matter of slow decline from then on. And that is completely wrong. We now understand that the brain is really activity dependent, and we're building our brain. It's changing all the time based on how we use it. And it's not just the genes. We think it's how you live your life that's going to have a big determinant on your risk of this disease.
So these things are about building better brains, either actively through things that you do or indirectly through the kind of person you are, I presume.
One of the things we've looked at a lot is psychological distress. That is the tendency to have depression and anxiety. People who were very prone to be distressed had about double the risk of people who were calm and easygoing, if you will. Another risk factor that probably works differently is conscientiousness. It refers to the tendency to be sort of self-disciplined, under control. People low in this trait were about twice as likely to develop dementia and Alzheimer's disease as people who are high in the trait. Even though it's a personality trait, and we think of personality traits as relatively stable, there's a tendency for conscientiousness to grow throughout the adult lifespan.
And these people must be fairly conscientious. That's from the religious order study, right?
Well, we're frequently asked that, but in fact, the average level of conscientiousness in this group is almost exactly the same as in the general public. Prior to our research, there have been a number of groups that had shown that social isolation was a risk factor for dementia. We found that it's not just being alone that puts you at risk for dementia, but it's also feeling alone. So it really suggests that there's a lot of this disease that is outside plaques and tangles.
So this is a nun from Dubuque She died, she was about 80 years old. She had 18 years of education, and she clearly had a mild dementia at the time that we last saw her, probably Alzheimer's disease.
And let's see what it did show here. And you can already see the abnormality of the plaques. All of these black structures here are plaques within her brain.
Okay, so the first case, pretty typical Alzheimer's disease. And the second case, she was over 90 years old. She had 20 years of education, no decline over the years, over 3 years of follow-up. What do you think we're going to see here?
Well, it looks like she should have a somewhat normal brain. She indeed did have Alzheimer's disease pathology in her brain. She has a significant number of plaques, and enough plaques to confirm a diagnosis of Alzheimer's disease. So her brain looks very similar to the nun that we saw that did have dementia. And yet this was somebody who was older, functioning pretty well, and had really no reason for us to suspect that she had Alzheimer's disease.
So the basic idea behind cognitive reserve is that two people with the same amount of Alzheimer's changes, one will have memory loss and dementia, and the other one could be completely normal. So what underlies that? Why is it that pathology of so much in one person is having such devastating effects, and the same amount is not having that effect in another person? And so the idea of reserve is that pathology is developing in the context of an individual that is somehow protected. All of these things about how you engage with the world have real scientific basis now that doing these things might be protective. One thing we've looked at, for example, is social networks. How many people you feel comfortable with talking to and confiding in. How many people you, I would say, know intimately and feel comfortable with. And the larger that network, the less likely you are to decline and the less likely you are to experience clinical Alzheimer's disease. So I actually have some data. This woman in her 90s had a social network of 10. So the average is 7.5 in the study. She had 10, and the other one had 1. The cognitive activity score in the one that was not exhibiting dementia was higher than the other one. And so the findings are pretty consistent with what we see when we do the study with everyone, and that is the one that seemed to be to tolerate the pathology had a vastly larger social network and was less prone to distress. If you're losing cognition, something has to be going wrong with the brain. It's not functioning properly. And so if something, proneness to distress, social networks, loneliness, depression, whatever it is, if it's causing memory loss, it has to be doing something to your brain. If you could identify that pathway and we can intervene with them, we could increase your reserve. And so despite the fact that you develop Alzheimer's changes, we think you may not get memory loss.
All right, now, how are we here? If the weather were nice today, I could go to the park.
You understand?
Yes.
All right. Was this word in your...
Growing.
Growling.
Growling.
Growling. You know what growling means?
Growling.
Do you know what growling means?
Yeah.
Make a noise,
yeah.
Right,
Okay.
And growing means to get bigger.
If you take your brain with you into your 10th decade, as you enter that 10th decade, you want the most robust, efficient, rapid decision-making, multitasking brain that you possibly can have, because that's what's going to allow you to maintain your cognition despite all the other things that's going to happen to it.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
This is my normal route to work. This is the way I drive every day to work. And yet I got a feeling that today is a day that actually changes the whole of my life. If it is something that's going to help millions of people, it will certainly be the first drug that I've been directly responsible for. And the fact is, I don't know what's going to be the result.
Word of a new approach to Alzheimer's. Until now, doctors have only been able to treat the symptoms of the disease, but experiments are underway on a new class of drugs that might stop the disease in its tracks.
All right, so here we are. We've got the next box for the next 3 months. And of course, the last package is always in case of a snowstorm.
Researchers are enthusiastic because fluorazan is the first drug that strikes at what might be the underlying cause of the disease as opposed to treating symptoms. Fluorazan is being developed by Myriad Genetics.
The result of this trial, whatever it is going to have a huge impact on people's thought process about treating Alzheimer's disease.
The most critical step in developing drugs is finding the right target.
One of the characteristics of Alzheimer's disease is the buildup of an abnormal protein called beta amyloid, which is associated with the destruction of brain cells. Researchers think fluorazan works by reducing the amount of amyloid plaque.
It's not just that amyloid causes Alzheimer's disease, it's how much amyloid do you need to have in your brain to cause Alzheimer's disease. And it's like a bank account, if you want to see it this way. The amount of money you have in your bank account depends on how much money you put in and how much you take out. And that's what it is with amyloid. Everybody, including myself, is making a certain amount of this toxic amyloid. But we don't have the problems because we're also clearing it. But as you get older, your ability to clear the protein goes down. And so what you end up with is a net accumulation of the amyloid in your brain, and then all those bad things happen.
I've been aware of a host of studies in the last decade and a half that suggested that nonsteroidal inflammatory drugs, NSAIDs, or the drugs that you can buy over the counter that lower inflammation, such as ibuprofen, Advil, etc., that these drugs have been known to lower the risk of Alzheimer's disease. That is, in individuals who take these drugs for more than 2 years, there appears to be a lowered risk of Alzheimer's disease. We saw that, in fact, non-steroidals had a very strong effect in reducing A-beta 42 levels.
These drugs at high concentrations, independent of their activity as anti-inflammatory drugs, all have risk. They all have adverse effects. And the Alzheimer's disease population is a particularly frail one. So that it looked suddenly like, well, this is not going to work. We can't give high enough doses of the drugs to do what we want in the brain because there'll be too much stomach upset. There might be gastrointestinal bleeding. There might be adverse effects on the kidney. It's not going to work. Well, that led to searching for an alternative form of an NSAID.
I don't know what happened behind the closed doors of Myriad Pharmaceutical, but certainly to someone from the outside, it looked like we kickstarted their Alzheimer's disease program, because shortly after we approached them, then their Alzheimer program really was formed.
We've lived with this compound for 8 years now, and when we got it, it was about understanding it in cells in culture, neurons in culture, and now it's in people.
If Flurazan does what it's supposed to do, it will slow down the rate of progression of Alzheimer's disease. You can't see that by treating 12 or 20 or 100 individuals because the natural rate of progression varies from one person to another. So in order to do a randomized controlled trial that is definitive, that will clearly show what the impact of the treatment is on the disease, you need 1,000 participants and you need to follow them for at least 18 months. That's what we have figured out. Well, that's a phase 3 pivotal trial, and that's the trial that's going on now. It's 1,600 people followed for 18 months. Do you jump into a trial like that without knowing that it's going to work? Well, you don't have too many options here.
How are you doing?
Oh, so good to see you.
Nice to see you too.
I miss you when I don't see you.
You do?
Yeah.
I don't know about that. Come in and have a seat. We're going to do a quick check.
Well, clinical trials are now the gold standard for determining whether any medication works. Phase 1 usually is just a very small group of people who get tested for safety. And phase 2 is a slightly larger group of people, say 100 or 200, who get tested for safety, dose finding, and any side effects that might be popping out, and also to give people a feeling for whether the drug is working, but may not have enough people to determine for sure whether it's working. And then phase 3 is the phase that's required for FDA approval. And that phase usually has a large number of patients who are enrolled. And in that phase, you take a good hard look at the efficacy, whether it works or not, and at the side effects or safety profile.
Any changes physically? Anything new?
It's really difficult to say. It's more subtle changes. He's not able to make decisions like he used to. For instance, in choosing clothing, things like that.
Okay. If things go well, you'll be able to stay on the medicine, and then hopefully this will be approved for the general public down the road for treating this disease. Okay?
Well, that's what we're hoping and praying for. All
right.
We want to see this work.
We'll take those prayers.
We discovered, much to our surprise in the phase 2, that some patients actually improved. Now, you don't automatically expect that. It's a progressive disease. You're treating the cause of the disease. You know, the best we hoped for was that we could stabilize them. They don't get any worse, but they don't get any better. But to our surprise, we saw some patients who actually got better. And so one of the questions we're asking in this phase 3 trial are did any patients get better, and what percentage of them got better? Did any patients on placebo get better? Did they all decline?
Thank you so much. Thanks for coming. We'll see you in 3 months.
Okay.
You know, I'd be happy with it to live with this the next 10, 20 years if it wouldn't get any worse. I'd be very happy with, I mean, I can deal with this.
Oh, yes.
Oh, hey, thank
Okay.
you. Yes.
And I'll let the nurses know you're here.
Okay.
Okay?
Fine. Ms. McCourt?
That's right.
Hi, I'm Dr. Stern.
Hi, Doctor.
Nice to see you. I'm not sure if you remember the last time we did this. We gave you a whole bunch of different questions to see how your memory and other things are going. Great. I'm going to say some words, and I want you to try to remember them. Potato. Toothbrush. Broccoli. Screwdriver. Tangerine. Banana.
Banana. Screwdriver. Toothbrush. Broccoli. That's about all.
I'm going to show you a picture, and I want you to tell me everything that's going on in the picture.
What looks like a barbecue with the mother and father and the 2 children and the dog, and that's about it.
Okay. Do you remember that list of words I just told you?
I hope I do. I don't know if I do or not. Banana.
Yeah.
Potato. You're right out of my head.
One of them was a tool.
I know, but I can't think of the name of the tool.
That's great. Thank you very much.
You're welcome.
I'm all done.
Okay.
Dr. Green,
Haven't seen you in a long time.
how are you?
Long time no see.
ya. Hi, Ms. McCord. Good to see you
Good
again.
to see
How are you
you.
feeling today?
Good.
Great. Did you notice any changes, improvements while she was on the study drug?
Yeah, in the beginning, she seemed to perk up. She was reading books. She stopped reading books. She started reading books again and started getting involved with the neighbors and things like that.
Has this been an okay experience for you?
Very, very much so.
So today, whether or not you've been on the study drug so far, which neither you know nor we know,
Okay.
today you do get to start on the
Okay.
open label drug, meaning we know it's the active
Okay.
ingredient. The Flurazan clinical trial is exciting because if it's positive, it may be the first positive disease-modifying trial for Alzheimer's disease. We've had drugs for a while that have been able to symptomatically treat Alzheimer's disease. They've been on the market, but we haven't had any drugs that have been thought to actually slow down the biological progression of the disease. So the development time of the drug plus the clinical trial time probably adds up to somewhere between 5 to 7 years at the very best in order to get a brand new drug to market.
Every day. Eating bread by 3. Here, you say you belong. 2 in the morning, 2 at night. You don't forget that Flurazan. You don't forget it. Only this is the real McCoy. Along with all the other medicines that are in here, this is a new one. I just kind of feel empty. For a long time, I didn't really believe that I had it. And when I got the medicine, it helped me feel better.
You don't know until I have a chance to analyze it and run it through the computer, and then take all the other people that were on it and run that through the computer to see what you come up with. So we don't know what we were on. If you got 0, that's better than nothing. To have someone doing something and trying out something. I mean, it took Edison 5,000 experiments to make the electric bulb, And the guy says to him, "You mean you did 5,000 things wrong before you invented the electric bulb?" He says, "I found out 5,000 things that wouldn't make an electric bulb." And that's just about the way I looked at it.
The scientific question that we are asking with the Fluorizan trial is whether Fluorizan slows down the progression of Alzheimer's disease in patients with mild forms of the disease. So with a larger sample, if those trends continue, and we see those same trends in the phase 3 trial that we saw in the phase 2 trial, we will have enough sample size to achieve statistical significance, and that will be the definition of a positive trial.
The fact is that I've been working in the pharmaceutical industry for over 25 years, and I've actually never had a day where I'm going to find out whether we have a drug that's going to help millions of people or whether in fact it's another compound that just gets thrown onto the heap. But I think we've probably spent $200 million to date, and we don't know the results. Once we know the results, we'll spend about another $300 million preparing to get the product onto the market.
Today's a bit of a milestone. This will be our last steering committee before the release of U.S. Phase 3 trial
If not, it's
results.
bad news.
But we will continue to meet as a steering committee going forward. We're planning on hiring 39 positions. Moving on to the marketing.
Are we putting the number on the press release? No.
No, on the website.
So is it going to be right on that front page, right as you come in, it says, for more information on Fluorizan, here's a number? I mean, that's what I would think.
I think that's
Oh, and fingers crossed, everybody. We have a lock on the door on the outside of this room. Nobody's allowed to be in this room by themselves. These are individual patients' records from the trial. The FDA is going to come and audit this. They're going to make sure when we say to them, this is the data, they don't take our word for it. They're going to come in and they're going to go through all these records and check for themselves. One of the problems with doing an Alzheimer's trial, as opposed to, let's say, a cholesterol, if you take a statin drug for treatment of high cholesterol, you do a clinical trial and you take a blood sample from the patient and you measure the cholesterol level in the bloodstream, and there you are. That's the answer to your outcome. So it's a very objective outcome. The problem with an Alzheimer's trial is that you can't take a brain sample from a patient. So your outcome is the outside manifestation of their brain, in other words, their ability to remember or how they behave. And that varies from day to day. I mean, it's always hard to deal with, so we'll see.
So you're headed out?
Yeah, so you know where my house is?
I do.
Okay.
Yes, I do.
Then I will see you there in about 10 minutes.
Okay.
Or so?
Yes. Drive carefully.
Yeah. There's a lot of Wall Street investors gambling on whether Fluorizan is going to work or not, and it's going to make a huge difference to the share price of Myriad in one direction or the other. And we are concerned to keep the data as quiet as we can until such time as a press release comes out. And it seems hard to believe right at this moment that in 15 minutes time or so I'm actually going to get to hear what the outcome of the trial is. There's Ed. Wow, there's some of the team are here. So how are you feeling right at this minute?
Pretty anxious.
Yes.
It's hard to believe the moment is about to occur, isn't it? After all these years.
It's been a long time coming.
Boy, hasn't it just?
Hi guys.
There he is.
I have to formally shake your hands. Bob, it's great to see you.
Good to see you, man.
Thanks for coming in.
Bob. Hi, good to see you.
How are you,
Bob.
Ed?
Thanks for coming.
We would have said hello to these
Good.
guys.
How you doing?
Yeah, let's just get it over with. I can't stand it any longer.
I know.
I'm sure. This has just been an incredible experience, and given all of the data that we've had up until this point, everything that we've looked at from the bench science in the lab to the clinical data that we've collected, given in that context, I think these are extremely surprising and just something that I never would have expected. So with that, what I think the most important thing to sort of communicate right off the top is that in terms of the primary endpoints of the study, we did not make statistical significance on either of the two.
Wow.
Like I said, I think a shocking result given where we were up until, from my point of view, yesterday, with everything that we learned about this drug and this program.
It's shocking.
It is shocking.
It's
Very
a
shocking.
definitive answer. I'm very, very sorry it's not the positive answer, but it's a extremely well-run study, extremely powerful study, and it gives us a clear-cut answer about this avenue.
Yeah. It's unfortunate the results came out the way they did, but that's clinical trials, right?
Okay.
Thank you.
Drink. Hey, well, sit and relax for a few minutes or whatever you want to do.
Thanks.
Walk in the garden. Think about things.
That's a shocker, I have to say.
Yeah.
Unbelievable. What are the implications for the amyloid theory, you think?
I don't know. I think we keep waiting to find a treatment that verifies or shows proof of concept of the amyloid hypothesis. And there is not yet one that's definitive. So this one falls by the wayside, even though we know that the compound does something to the amyloid system.
Yep.
We don't know whether it didn't do enough or whether there's something fundamental in the amyloid hypothesis that makes treatment not as effective as we might have thought.
It does probably make people want to think about the correlation between laboratory findings and human findings.
So where does that leave us with the epi data from the non-steroidals?
Yeah, I think it's going to stimulate a lot of scientific discussion and hopefully will advance the field.
We'll go ahead and tell the board, well, yeah, try and tell the board tomorrow, but I think, and then press release Monday and be done. I think we probably just need to get it out of the way tomorrow. But it's a dashed hope for a lot of people, unfortunately. So.
All those caregivers are going to be devastated.
Yep. Yep. I just want to know why, because it defies everything I know. I mean, when you have all those independent observations from the phase 2, time to psychiatric events, that kind of thing, how can you go from that to no effect? I understand it.
I want to know why too. I want to know why you can see that kind of change in the basic science in the amyloid, and we so believe that the amyloid theory is correct, and that impacting that will influence the course of the disease, and there's data that you can impact it in humans. Every step of that logical chain.
I know.
And then the clinical trial is negative. So it's very exciting but somewhat bittersweet to report to you this, the largest phase 3 trial in Alzheimer's disease ever completed with the team from Myriad Pharmaceuticals. This is a well-designed and we think well-conducted study of patients with mild Alzheimer's disease. Unfortunately, there was no efficacy demonstrated between the placebo arm and the treatment arm, and the drug was generally well tolerated. We're very proud of the fact that this was one of the first clinical treatment trials for a disease-modifying compound, and while in technical clinical trial parlance, the drug failed, we believe that the trial was very successful in answering the question. Thank you.
This is the second International Conference on Alzheimer's Disease news briefing this year on therapies research.
Hi, Victoria Elliott, American Medical News. This question is for Dr. Green. If there are any theories as to why, since it did do so well in the earlier trials, why it didn't work at the phase 3 level?
You know, obviously we've tried to think about why it is that the phase 2 results were so encouraging, and I think it's honestly not clear. The most likely reason is that in a small sample size, as is always the risk in a phase 2 trial, there is a chance, by chance, that you will see results that don't turn out to be valid when you use a larger study. If you sort of think of the hierarchy of possibilities for why this didn't work, you can indict the entire amyloid theory, which I think is not a possibility. I think we ought to be pursuing amyloid therapies. You can think about whether this particular drug got in at an appropriate dosage and got enough CNS penetration, which is a possibility. And then as you're hearing from many people, the question of a therapeutic window, when is the right time to try a drug, is often very difficult to determine. And with many compounds, we have the feeling that the earlier we try them, the better.
What this all is telling us, though, is that we've got to go to earlier stages. We can't go to the mild to moderate Alzheimer patient. We need to bring our trials up to the mild cognitive impairment, if anybody can agree on that definition, and even to the earlier stage when you look at the risk factors for patients. That's the time to treat. It's before the pathology gets some steam.
What we need to do is not be studying the already demented. One of the things that I think the amyloid imaging is beginning to show is that there's this, so you start off, you have a little bit of amyloid, and then it just seems to flatten out, okay?
You're too late.
And you're too late there.
Correct. I mean, so our strategy then now is to say, we're going to try and look for certain markers, okay?
I think biomarkers are getting to the point where we're going to have what we need to do these earlier studies, whether it's secondary prevention or primary prevention.
I agree.
And I think that's exciting.
I agree.
I think that, there is tremendous enthusiasm, and that's what's going to get us there. We're going to get clues from partially efficacious trials and clues from trials that don't work out. We're going to get clues that are going to take us closer.
From a trial doesn't work perspective, it's terrible for us. I mean, it's a huge amount of work and a lot of disappointed people. But on the other hand, in the scientific literature, science moves forward, and it does, ironically, make a cure of Alzheimer's disease that much closer, but it just isn't going to be from this drug. So you've got to look at it that way.
Have you noticed any difference? I mean, do you feel like you've done better this time, or do you think that you...
I think I could have done better.
You think you could have done better?
Yeah.
There's been an exponential increase in our understanding of what causes Alzheimer's disease, how we can target it, and how we can treat it.
It really is miraculous that in a short period of time, which is really 25 years, and we know a lot about the disease.
We are at the brink of controlling one of the major diseases affecting world health.
We do have the research scientists, we do have the knowledge, and I think we can beat Alzheimer's disease.
Alzheimer's disease has been around forever, but in the last century or so, the population has been living long enough for Alzheimer's disease to be a significant problem. The disease was given its name just over 100 years ago. The next 3/4 of a century, taking us into the 1970s, really didn't see any attention toward the possibility of treating this disease. When I was in medical school, we spent no time at all on the disease. Just wasn't important because there was nothing that could be done about it. And it was really in the 1970s and then into the 80s that that thinking started to change. This disease is not something that's part of getting older, about which there's nothing that can be done. This is a specific disease. It has a specific pathophysiology, that is specific abnormalities in the brain that cause the memory impairment, and that we, the healthcare community, can treat those abnormalities and improve the situation. So the first small trial in 1985 launched a huge effort on the part of NIH and the academic world, as well as the pharmaceutical industry, that led to the approval of the first treatment in 1993, a cholinesterase inhibitor, Tacrin. Now we're at the point where we think we understand the triggers for the disease. There are two primary pathways. The tau pathway. Tau is a protein. It's a natural substance. It becomes abnormal. It's necessary for normal brain cell function, and it becomes chemically changed in Alzheimer's disease so that it clumps together in the tangle. The plaque, we now know, is made up of amyloid peptide. Amyloid peptide is a fragment of a normal protein that we have in all the cells of our body. But again, there are chemical changes that take a normal protein and cleave off a piece of that protein that happens to be highly sticky, and it clumps together and sticks to the brain substance and interferes with brain function. Now we believe we can target our therapy to one very specific step. If we can control the inciting event, the driving event, we can bring the disease under control. We can move from the stage of modestly improving symptoms to actually slowing or even halting disease progression, and ultimately we'll be able to halt the process before the disease occurs. Things have gone awry with the amyloid pathway and the tangle pathway long before memory impairment is apparent. And we are now learning that we can see those abnormalities using biomarkers, using measurements of the amyloid peptide in the spinal fluid and imaging the amyloid peptide in the brain using PET scanning. We can see these changes before there are any symptoms. In the case of Alzheimer's disease, you can't look at someone and see, well, the disease is at its 2-year point or 3-year point. So the first thing is you need accurate ways of assessing the disease. So when you have a treatment that slows it down, you can tell. That turns out to be a huge problem, one of the biggest problems that we face. But we're all working together. We're working together to define readily available methods for tracking the course of the disease. We believe that we can use this understanding and use the new methods that are continuing to be developed today. We will be able to halt the progression of the disease in people who have Alzheimer's disease. We'll be able to halt the progression of the disease even before the point of diagnosis today. And ultimately, the future that we see is that we will be able to assess risk in normal individuals as they age, and we will be able to control that risk at a molecular level. We'll be able to monitor and control the cascade of events that, if left unchecked, would lead to Alzheimer's disease. Drug development, it's a process that takes tens of millions, sometimes over $100 million. It starts with basic scientists in the laboratory that define the targets, find out which enzymes are involved, and we move then from the biological scientists to the medicinal chemists that design the interventions, that design the drugs, and the synthetic chemists that make those drugs. And then we need all of the biologists to test the safety of these interventions and their efficacy in model systems, in cells, in dishes, in animals. Then we need the pharmacologists to translate these treatments into human studies. We will never have a treatment without testing it in people with the condition. It's an essential part of the process. And so, no matter how rapidly the science moves forward, no matter how many terrific molecules are created, no matter how much evidence there is that we understand the disease and we know how to control it, we will not get a drug to market without large-scale clinical trials that prove that they work and that they are safe. There are many benefits apart from being part of the process of developing better treatments. The families learn about the disease, they get the very best standard care, they get the greatest attention, they are able to learn about everything that's going on in the field as a byproduct, as a benefit of participating in clinical trials. It's in a nurturing environment and it's a positive experience, but it is a big commitment. People agree to take a medication that may have benefit but may have no benefit and may have risk. These are brave individuals who are participating, and they're participating because they understand that there's no other way to move forward, that without their generous participation, we're stuck. We don't make any progress. The participants, they agree to take either a placebo or an active treatment. They agree to have brain scans. Some of them agree to have spinal taps. They're very generous. One of the best pieces of news is that there are many clinical trials now with very promising agents for Alzheimer's disease, dozens. They're being conducted by the Alzheimer's Disease Cooperative Study, the ADCS. They're being conducted by the pharmaceutical industry. They're being conducted in this country and in other countries. We are studying both traditional type drugs, novel molecules developed to hit a specific target, and natural and health food type interventions. We are studying DHA, which is an omega-3, an ingredient in fish oil. It's a natural product. Many people are taking it because they think it's going to help. We are trying to determine whether, in fact, it helps and is safe. We think there is good evidence. Not good enough to recommend it, though. Good enough to definitively test both safety and efficacy of sufficient doses to potentially have a favorable impact on the brain. That's what we're doing with DHA. Similarly, we have studied antioxidants and herbal extracts, and we find that some have benefit and some don't, and we find that some have unexpected risk. There's a general thinking that if something's natural, it's good for you, and if something is synthetic, it's bad for you. But in fact, the worst toxins we know are natural compounds that are poisons that come from plants, and some of the most effective treatments we have are entirely synthetic. That's not really a fair appraisal of risk. An herbal treatment can be lethal, and an herbal treatment can be beneficial. There are certainly natural extracts of herbs that have potent beneficial medicinal qualities, but they have to be tested both for effect and for safety, just like any artificial molecule. For the person who's in middle age, as I am, and who has a family member who's had Alzheimer's disease, which I do, it covers most of middle-aged people, who's appropriately worried about the risk of Alzheimer's disease in late life, what's the outlook and what can they do now? Well, what they can do now is lobby for more support for drug development for Alzheimer's disease, because I believe that before we, who are in our 50s, enter the age of substantial risk, which is our 70s, these treatments are going to be proven effective, the ones that we're testing and developing now, and we will be able to actually control risk through interventions that interrupt the amyloid cascade. That's what I think is the best approach. What many people are saying, okay, yeah, you're trying to raise more money, fine, but I want to do something to my lifestyle now that's going to help. And what I would say is we don't know. There are no good studies to justify any firm advice, but nonetheless, we give advice. We give advice that's based on everything we understand about the relationship between lifestyle and environment and Alzheimer's disease. But I want to emphasize that everything that we have now is not enough to know that any of these things make a difference. But nonetheless, we think that it may be useful to have a vigorous lifestyle with physical activity, with cognitive activity, with social interactions, to keep learning as long as you can throughout life, as much as you can, to pay attention to general health issues such as blood pressure control, blood sugar control, to have routine health maintenance and do those things that we believe are good for cerebrovascular disease and cardiovascular disease, because we think they may be helpful for Alzheimer's disease as well.